upfront. So my name is Marius Causemann. I'm a postdoc at Simula, and I'll present some work with my supervisor Kent André Madal on inverse modeling of CSF flow fields. But I want to start with a very simple, small illustration of the very basic idea. And I think with the current temperatures out there, it's maybe easy to imagine you're in a plane above the Canary Islands up there, and it's beautiful weather. You're looking forward to going to the beach, but maybe you're actually interested in surfing there, and you want to know what's the wind speed, right? And from an image like that, you basically can't tell, right? So it looks good. Maybe one could see some small waves if there was a lot of wind, but this situation changes quite a bit if we had actually some clouds, right? Now we can actually intuitively have a bit of an impression of what kind of flow fields we have. So we see, for example, this beautiful vertex straight behind one of the islands. So if we have some kind of tracer or some passive thing that is advected by a flow field, we can actually say something about this maybe otherwise unobservable flow field. And in the brain, that's what I'm interested in, we are in a somewhat similar situation, right? So I think Andreas and Cecile have made a good case of why we are interested in CSF flow in the brain, and we are in a situation where using some MRI techniques, we can relatively easily image the CSF spaces themselves, so we can see where we have fluid. So basically all the very bright white spaces are CSF-filled spaces that surround the brain and fill the ventricles, the cavities that we have in the brain. But saying something about the flow in there is relatively difficult, especially if we're interested in the slow-moving net flow, because we also have a lot of pulsations, right? So typical methods that one could use to assess flow using MRI techniques, their signal will be dominated by pulsations that come from cardiac and respiratory pulsations. So, and that puts us in a somewhat maybe weird situation where we know that about 500 milliliters of CSF are produced in the ventricles in the brain every day, but we really don't know where it goes, right? So it's not a negligible amount of fluid, 500 milliliters a day. And I brought a few papers here from the biomedical community from the last couple of years, and there are quite a few new outflow routes for CSF being suggested, or people just saying this question has been unanswered for about 150 years. And then I want to come to a work of some collaborators of mine in Oslo that have come up with a new method to compute or image so-called T1 values and T1 maps in the CSF in humans. And the important observation here is that the R1 value, which is just the inverse of the T1 value, is roughly proportional to the CSF protein content. So here on the left, you see the T1 image, a structural MRI image overlaid by the R1 value of CSF in the brain, and we see we have kind of these blue low values in the ventricles. So there we know CSF is produced. It's somewhat, let us say, clean. It has a low protein content, and then it's much higher in many of the other CSF spaces around the brain. And that is basically the main idea. So maybe we can use these protein concentrations to infer what the CSF flow fields and the absorption sites are, a bit similar to we can look at the clouds and say something about wind patterns. So to do that, we need a few modeling assumptions. So this is a relatively simple model. So we just assume that protein is transported by is advected by a stationary CSF flow field that comes from CSF production and absorption at different points. And then we have clean CSF being produced in the ventricles and proteins being generated within the brain, and then they enter the CSF via basically the interface between the CSF spaces and the brain, which is the pile membrane. So the first thing that we did then is, well, generate a mesh and then subdivide the outer surface of our CSF domain which is basically somewhat equivalent to the dura mater, into potential absorption sites. So we have these 15 different colors that are the different absorption sites. And then we associate one flow field with each of these absorption sites and sum them up and have this advective flow field B. And then we model this with a stationary advection diffusion equation. We have the clean CSF coming in from the ventricles and protein influx via the pale surface. And then I have to admit I'm joining the club of people that just use the most simple approach. So this is just discretized with a P1 element implemented in FEniCS. And then this problem is advection dominated, so we add a pretty standard SUPG stabilization for that problem. And then you might ask, how do we compute these candidate flow fields? And to do that, we solve an incompressible Stokes flow equation for each of these potential absorption sites, where we have inflow via the lateral ventricles in like these cavities in the brain, and then we have a prescribed outflow on each of these patches, and then have a no-slip boundary condition on basically all the remaining boundaries. And then, maybe slightly more fancy, we are discretizing this with the BDM-DG approach, which gives us a locally mass-conservative flow field. And then here on the right, what you see is a streamlined visualization of one of these candidate flow fields, which corresponds to absorption at the anterior sagittal sinus. So this is kind of something that sits here on the top of the brain between the two hemispheres. And we do this for all these different absorption sites. This is surprisingly blurry at the bottom, but yeah, I think you see all the other flow fields. And then we go on to actually solve this problem. And we use a Gauss-Newton approach to solve this PDE-constrained optimization problem, where we try to infer these coefficients of these advective fields. And this is maybe a bit different from standard like large-scale optimal control problems, because we only have these 15 unknowns, so we can actually assemble these matrices involved explicitly relatively cheaply. And in that case, we can get the Gauss-Newton approximation of the Hessian by basically we need to solve for this n times, but since it's a linear problem, we can reuse one LU factorization and get this pretty cheaply. And then we use an active set approach to solve each of the quadratic problems at each of the steps. And then we see that this, in our cases, converge relatively quickly in about 20 to 25 steps. And then this is a setup where we try to recover this flow field if we just create a synthetic data set with only one single basis field. And see, this works really, really well. So here the target and the reconstruction agree quite well. And we did this for basically all these absorption sites, and then plot here the relative weight of each of these coefficients. And we see that this works really well. So we only want these diagonal entries, and that's pretty much what we get. This is at 10% noise level. And then we did the same thing and increased the noise level here to about to 50%. And then this still works relatively well with the some smaller deviations here from the diagonal. And then finally, we applied this to a data set that was published together with this, new MRI sequence to quantify the T1 maps. So what you see here on the target row here is the actual MRI data, and then the bottom one is our reconstruction. I think this fits quite well in some regions, maybe not perfectly in all, so we kind of underestimate the concentrations close to the brainstem in these lower regions, so that's maybe something to look into. But if we then look at our reconstructed absorption sites, we see that it's primarily located here around the the posterior and anterior parasagittal sinus. So these are these regions here between the hemispheres on top. And then we also have some contributions from the occipital lobes. So these sit somewhere at the back of the brain. And I think maybe for some engineers this is not very exciting, but I'm already happy that we are roughly hitting the right order of magnitude in this kind of first attempt, right? So we are in the order of maybe, what is it, like 300 to 400 milliliters per day being absorbed. We know it should be around 500, and I guess in this type of biomedical applications, that's a pretty good fit. And then maybe a bit on some future work. So one thing I want to look into a bit is kind of how reliable are these numbers that I am computing here, especially with respect to some of the modeling assumptions and parameters that we've chosen here. And then run this on more subjects. So we have a data set of 30 subjects, and some healthy and some Parkinson's patients, and then maybe we can also compare if we have differences in their CSF flow fields. And then I think maybe a bit further down the road, we could apply a similar idea to the MRI tracer data that Cecile was presenting earlier, which would essentially be somewhat similar problem, but then in a more complex time-dependent case. And yeah, with that, I want to thank you and look forward to questions and thoughts.
So are there any questions for the speaker? It seems like we've now had enough presentations on the brain so everyone understands everything, so it's crystal clear.
I'm also interested in reproducibility, and typically in medical applications it's like, we have some secret data sets from the hospital and so on, but this GONSO data set, that appears to be like an open data set.
Yes, so that is an open data set that is up on Zenodo, was primarily made by a previous PhD student colleague at Simula, Riseth, and yeah, everyone can download and play around with it.
But did that play like an important role, or because that means many people can use the same data, right? Could you comment on that? Was that?
I mean, clearly, if you don't have access to the data, then you cannot do this kind of thing. I guess I would have been able to do this without the GONSO data set because I sit at the source of this. But I think it is just important that we share this data and make it possible to for other people also to develop methods and work with this.
Yeah, I thought that was very positive.
Yeah.
If there are no more questions, I think we should thank the speaker again and all the other speakers.